Excellent. Okay, so going back just a little bit, we're going to talk a little bit about like amino acid metabolism, fat metabolism a little bit more, the ATP tally count, and kind of interactions between the anaerobic and aerobic energy systems. But really quick here, just something that I wanted you to know. We get fatty acids into the cell through fatty acid translocase, FAT, right? That's how we get it into the muscle cell. And then we actually get it into the mitochondria with these particular enzymes that I want you to know about. So carnitine, like acetyltransferase, and this is collectively part of the whole like carnitine shuttle. But there was another word for it that like I just want you to write down, carnitine palmitotransferase. So I said it last time. I figured I would put it up here today so that you could see how it's spelled. So palmitic acid, you know, that's like our 16 carbon like saturated fat. Now it is doing a lot of stuff like transferring acyl groups and doing different things with like palmitate, but like both of these terms are terms that are used interchangeably. They mean the same thing. Now, most of the time the acronym used for these is CPT. So not certified personal trainer, but carnitine palmitotransferase. That's how it goes. And CPT1 and CPT2 are the different ones. So CPT1 kind of like beginning to get us like to go through like that intermitochondrial membrane space, and then CPT2 gets us into the matrix. So CPT, I'll probably mostly refer to it as that if it ever comes up again in class. Okay. Next thing, just reiterating a little bit what beta oxidation is, functionally the process. Since acetyl-CoA is 2 carbons in length, we have to break down the fatty acid 2 carbons at a time, and that's functionally what beta oxidation is. Now, all of this, I obviously don't want you to remember all of this. I just want you to understand like the complexity of like the whole beta oxidation. Now, like glycolysis, I do want you to know a lot about this, but this I really just want you to understand and like appreciate a little bit more. Another thing in particular about this, in laboratory settings, if we isolate a mitochondria from someone's muscle or like whatever type of tissue, we would put it into like this like chamber effectively, and we can measure how much like carbohydrate versus fat that it has the capacity to burn. So the reason I'm telling you this is there's typically a relationship between NAD and FAD production whenever we're burning like glucose versus fat. And glucose, typically more NAD production versus FAD. Everyone good with this? Everyone good with that, right? In fat, it seems to be a little bit more FAD versus NAD. So how we like test that in like mixing chamber type of study, we would essentially put like an antagonist or shut down complex 1 in the electron transport chain, which accepts the NAD to virtually just see how much FAD that it can actually take up, and functionally fat production or fat oxidation. Is everyone good with this? That makes sense? Like, I don't want you to understand like the crazy complexities about that, just really that fat oxidation is more related to FAD. And does anybody remember how many ATP are produced per NAD and per FAD? Anybody? All good, write it down. NAD is 2.5, FAD is 1.5. So since fat is doing a little bit more FAD versus NAD, which one is more efficient in making ATP? What do we think? NAD. Yeah. I'm sorry, I didn't hear you, Sydney, whenever you were talking. Oh, okay, okay. So like, since carbohydrates produce more NAD relative to FAD, we actually get more ATP out of like carbohydrates for the amount of carbons in it. Everyone good with this? Right? That makes sense? Because 2.5 NAD, 1.5 FAD, and if the relationship is a little bit more FAD for fats, then we get less bang for our buck out of there. Everyone good with that? Okay? Cool. All right. moving on from there, like I like this picture a lot because it really shows more or less the cyclic nature of like how like a fatty acid like functionally like comes in right here. We take off a couple parts of it, and then then we get like a 2-carbon acetyl-CoA, and we just go through this numerous times until the whole fat is gone. So like if we have like 16 carbons, you know, like we go through it like 7 times or so. Is that right? Something like that. It's that's not really like much of a big deal for you to know, but really just beta oxidation is chopping off 2 carbons at a time because acetyl-CoA is 2 carbons in length. Hopefully that makes sense. All right, moving on just a little bit. These days, a lot of people talk about the ketogenic diet in many ways, and most people don't even know what ketones are. Does anybody know what a ketone is? No? No? Like not at all? Okay. So like there's 3 ketones in particular that I'm going to show you. There's one, beta hydroxybutyrate. Frequently it's called like BHB. And some biochemists don't even consider that a ketone for various reasons, but like functionally it basically is. There's another one called acetoacetate, which is a ketone, and then there's also one called acetone. So how we make these, and this is what I want you to know. We make it from fat. We essentially, in the liver, go through like beta oxidation so much that we get so much acetyl-CoA that there's a backlog in the Krebs cycle that we can't even like run the Krebs cycle because there's just so much acetyl-CoA. So right there, like beta oxidation, acetyl-CoA, we get a bunch of those, right? So acetyl-CoA, 2 carbons. More or less, what ketones are is the combination of 2 acetyl-CoA molecules to make the ketone bodies. Now, really the main reason for this is the blood-brain barrier has somewhat of a size limitation for like taking in energy. Is everyone good with this? Right? So pretty much the main reason that your brain runs on glucose is because it's a good size to like get through the blood-brain barrier. C6H12O6. Everyone good with that? Good? Good so far? Right? Now, can someone tell me the chemical formula for palmitic acid? Man, that's funny, right? It's okay, so like that's 100% going to be on the test now. Cool? Everyone good with that? Okay. C16H32O2. Now, just like hopefully we understand numbers, which one of these molecules is bigger? Yeah, palmitic acid, right? The 16-carbon saturated fat. So that can't cross the blood-brain barrier. So whenever we're producing ketones, it's whenever we're in a really low carbohydrate environment, meaning that you're not eating like glucose, rice, like whatever. And whenever we put all of like the acetyl-CoAs together, what we get, beta hydroxybutyrate, I want you to know this. It's, what is it? C4H. Let that out already. Can you all tell I didn't sleep a lot? Right? Okay. Yeah. Jeez. H8. Well, because you see it goes right there. O3. That is beta hydroxybutyrate. Okay. So the size of these molecules is really important. So our brain can't utilize palmitic acid because it's too big. Our brain is very good at using glucose because, I mean, it's in the Goldilocks zone. It's like not too hot, not too cold. It's just the right size. So we actually make ketone bodies to fuel our brain. Now, your muscles can use them too, but it's mostly to fuel the central nervous system. And since it's C4H8O3, that functionally is about the same size or smaller than glucose. So it can cross the blood-brain barrier and be used very readily. And in fact, a lot of people dispute which one the brain actually prefers to use. So I'm sure most of you have learned that like we need a certain amount of carbohydrates every day for your brain to work. Has anyone ever heard that? Right, okay. Like typically it's around like 120 grams of carbohydrates so that your brain works. And that's based off of your brain consuming 20% of like your resting energy expenditure. So like that's approximately where it comes from. We could backtrack that to 2,000 calories if we wanted to, but I'm already belaboring this point too long. But beta-hydroxybutyrate is such a small molecule, it can get into the brain relatively easy, and we can make a lot of ATP for your brain to work whenever we're absent carbohydrates. Is everyone good with this? That's functionally like why this works. Everyone good here? Yes, DJ.
So that BHB is a ketone?
Yes, yes. Yeah, it's like up here, BHB. Technically, the ketone bodies, beta-hydroxybutyrate, BHB, right there. And then acetoacetate and acetone, those are like the other two, more or less. Really, BHB, this is the only one that I even care that you know. Because it's like really whenever we're circulating things in the blood, acetoacetate is relatively unstable, and it's carbohydrates to acetone. So like something funny. Has anyone ever tried a ketogenic diet before? No? Nobody? Okay, so something that I think is funny about it. Has anyone like ever done a breathalyzer? I'm sure some of you have, right? Okay, so like whenever you do a breathalyzer, there's metabolites of like ethanol or alcohol metabolism that it's actually measuring. And the breathalyzer actually isn't that good at differentiating between different types of like molecules. So if you're on a ketogenic diet and you're breathing out a lot of acetone, you'll get a false positive for being intoxicated. Right? So that's kind of cool, huh? Yeah, so like, I don't know, I think it's cool. So like myself, they've done breathalyzers on me quite a few times because I'm friends with cops, right? That's like whenever I'm on a ketogenic diet, then yes, I can actually hike that up really high and like make it to where like I'm well past the legal limit. So yeah, I don't know. That's yeah, I probably shouldn't share so much about myself. So, all right, moving on. Okay, that's all I want you to know about ketones, more or less. Just kind of like how it's put together. It's from a lipid. Like we get too much acetyl-CoAs, we put two of them together, and then more or less we make beta-hydroxybutyrate. And that's all done in the liver, And it's to like go to the brain so that the brain has access to energy whenever we're deprived of glucose. So, man, that's a terrible looking 6. But all right, that's why I type things. Okay, now amino acids. There's a little bit that I want you to know about this. So branched chain amino acids. Like I've already talked that those are the main 3 that are important for exercise. So like what are they? Leucine, you really need to know this stuff. Leucine, isoleucine, and valine. So like go back if you don't know how to spell them. The metabolism here is with something called BCAT, branched chain amino acid transferase. And within that BCAT enzyme, this is where we see another one of our really big deal B vitamins, like the energy like vitamins, right? The B complex. So vitamin B6 is really important within BCAT. So if anybody's ever taken like different like branched chain amino acid supplements, almost all of the time they have like 100 to 1,000% of the vitamin B6 that you need in a day. And the reasoning for that is right here. Like right there, because it's like B6, it's a really important, I don't know, cofactor, coenzyme, whatever the hell you want to call it, in branched chain amino acid transferase. So it aids in that metabolism, more or less. Is everyone good with this? Right? Okay. So overall, the branched chain amino acids, they go down, we break them apart. There's a couple of other things, really just that first BCAT is what I want you to know. Then they go into intermediates in the Krebs cycle. So mostly it's somewhere like near the bottom, like succinyl-CoA. But I'm fairly sure leucine can even be converted into acetyl-CoA. I'd have to go back and look at that though. But I want you to know that branched chain amino acids can be converted to intermediates in the Krebs cycle, or citric acid cycle, whatever you want to call it, so that we can derive energy out of them that way. So deriving energy out of these is exclusively aerobic. So we have to do everything in the mitochondria and get energy that way, more or less. So again, oh yes, Sydney.
So all the branched chain amino acids, they can be converted into intermediates in the Krebs cycle to produce energy.
Yes. Yeah, to produce energy aerobically. Yeah. So typically, the type of athlete that would benefit the most from taking a branched chain amino acid supplement would be a long-term endurance athlete. Because if you're lifting weights or sprinting, if you're breaking down energy like aerobically, run faster or lift harder. Basically, that's kind of the take home there. Now, I can't remember, I was talking to Quinn about this, but has anyone ever seen the supplement HMB? Like, they actually put HMB in a, does anybody know, like, the Ensure, the old people drinks, so that people don't lose, like, muscle mass, sarcopenia, and all that type of stuff? So, like, they've started to put HMB within those. And what HMB is, it's a metabolite, or a couple of steps down the road, of leucine. So there's an idea that leucine, if everybody remembers AKT mTOR or PKB mTOR, right? There's an idea that leucine has the largest effect on phosphorylating mTOR. So essentially starting the whole muscle protein synthesis process. Now, the metabolite of breaking down leucine, like through various things, like I don't care if you know any of this stuff, is HMB. So like beta hydroxy, beta methyl butyrate. Doesn't really matter if you know that, but just HMB. It seems like it might be a little bit better at stimulating mTOR. So old individuals going through sarcopenia, like sarcopenia just means losing muscle mass due to age, more or less. If we have them consume a lot of HMB, which might be better than leucine at stimulating mTOR, might be, like I'm hedging my bets a little bit there. They might lose less muscle mass over the course of their life, and then like, I don't know, they will be all like jacked like old dudes and ladies and won't slip and break their hip on ice, right? Like more or less that's kind of the idea there. Is everyone good with that? Right, so just kind of like fitting in where HMB works there. Okay. Now to the ATP balance sheet, and I promise we're almost done with exercise like with bioenergetics. I mean, sorry. Okay, so this is like what Josh was thinking about the other day. How many ATP do we get from glucose? I want you to know this. One glucose molecule, C6H12O6, gives us 32 ATP. Now, something I'm going to say now, and it's not written down, so like write this down. What if we start from glycogen and not glucose? So if we go back and look at bioenergetics, glucose, that's circulating in your blood. Then it gets into the cell, that sugar trap step, hexokinase. We lose 1 ATP there. Glycogen is already stored within the muscle. So if we start with glycogen and we don't have to lose that 1 ATP, hexokinase, how many ATP would we get if we started from glycogen? 3. Yeah, 33. 33, yes. So write that down, everybody. 33. 33. So, in a sense there, glycogen is actually slightly more efficient, right? Is everyone good with this? Right? Now, the problem with glycogen is we have a limited capacity. So the body wants to effectively protect that until it has to use it. And we're going to get into that more in exercise metabolism, but like moving on from there. All right, so recent research, 1 glucose, 32 ATP. I want you to know where all of them are coming from. So here, like how I talked about like the energy generation phase, giving us like 4 ATP in glycolysis. Now we're talking more like net ATP. So in glycolysis, and this is in your book, so like check that out. In glycolysis, we make 2 ATP net. 2 NADHs, and 2 NADHs, if we multiply that by 2.5, that equals 5. Is everyone good with that? Next thing, the one almost everybody forgets about, the pyruvate dehydrogenase reaction, which is taking pyruvate to acetyl-CoA. We actually get 2 ATP from there, because if you recall, glucose, at some point, like after like the aldolase reaction, the one that I didn't care if you knew, right before the glyceraldehyde 3-phosphate dehydrogenase reaction, we have like 2 branches of it, right? Everyone good with that? So we get 2 pyruvate from the breakdown of 1 glucose molecule. So pyruvate dehydrogenase, we get an NAD there, but it happens twice, so just like keep that in mind, right? So that's going to be another 5. At the bottom of the Krebs cycle, we get 2 GTP or 2 ATP. You can write that, like that's fine with me. Like the difference between GTP and ATP, I don't find that compelling really. And then in the Krebs cycle, 6 NADHs. So like the figure I showed you from the Krebs cycle, like all the dehydrogenases that I wanted you to remember, there were like 3 of those functionally, like that yielded NADHs. So if we have to go through like 11th commandment, everyone remember this, right? 11th commandment, the Krebs cycle always goes twice. 3 NADHs means 6 NADHs. And there is 1 FADH within the Krebs cycle. So then there's 2. If we add all of this together, we get 32 ATP. Everyone good with this? That makes sense? Now, this is a lot to study, so like, you know, like study it. That's, I mean, pop quizzy type stuff, maybe. I don't know. We'll see. We'll see. Now here. I'm not sure in your notes, like this might not be filled in. But this is functionally saying the same thing that I was saying on the last slide. So 2 ATP up top from the NADHs, 2 of them, we get 5. And then 2 NADHs from the pyruvate dehydrogenase reaction. And then in the Krebs cycle, we get like 2 ATP. It says GTP in your book, but like truthfully, the more I read about it, it seems like GTP is made in the liver, but ATP is made in the Krebs cycle of like muscle cells. But that's not a particular note that I care if y'all know about. Now the NADHs right there, getting 15 ATP out of that because, well, like 6 times 2.5 is 15. Now, like just memorize that if you don't want to do the math. FADH is 2 times 1.5 equals 3. Hopefully everyone's good there, right? So then we get a net yield of 32. Okay, so there, this right here, there's only really 1 point, and this very last bullet is the main thing I want you to know about this. Most of the energy, like most of the chemical energy that like we actually take out of like different like macromolecules, and whenever I say that I mean glucose or fat, is actually lost as heat. So overall, humans, we're only about 34% efficient at like actually converting like chemical energy into mechanical energy. Now, a lot of people think that's wasteful, but since we're like, I don't know, warm-blooded creatures, like we're not reptiles, like that's probably kind of a good thing, right? Because, yeah, I don't know. Yeah, that's really all I have to say about that. Yeah, everyone good with this here? Okay, good. Like this thing about 7.3 kcals per mole, that's like we're not going to be doing stuff with that, so I don't think it matters all that much. Okay, control of bioenergetics. We've already talked about some rate-limiting enzymes. So PFK and isocitrate dehydrogenase being the main ones that I want you to know. But really, what rate-limiting enzymes overall are, are just a point in like a set of chemical reactions that control how quickly that like set of chemical reactions will occur. What's the example people always use? Like if you've ever been on the highway, like if they close off one lane and they make it to where like there's only like one lane or so, like that choke point is virtually like the rate limiting enzyme step. So like if you do something and open that up, then traffic flows a little bit faster. Everyone good with this? Hopefully you've heard something like this before. If not, it's like it's an analogy. Take it or leave it. Doesn't matter. I never think about it that way. But like there's various modulators that seem to like open up like that choke point in the reaction, and typically they're energy related. So if we have a lot of ATP, then we don't need to go through the bioenergetics like systems very quickly. But if we don't have a lot of ATP, we kind of do, right? So more or less within this, ATP is like one of our primary products of all of these energetic pathways, because like we're trying to get energy to move, right? So if like we're at rest right now, we probably have a lot of ATP, a lot more than we need, because no one is in here at the moment. We have a lot of ATP that's going to go back, find a rate limiting enzyme, and slow it down. So just about all of you, your PFK activity is probably really low at the moment. Now, like me being up here, since I'm moving around a little bit, it's probably a little bit faster, but it's neither here nor there. Hopefully that makes sense. Now, this right here, there's various stimulators and inhibitors, like that's what I've been talking about more or less. And I want you to think about this a little bit. So in the ATP-PC system, there's only one reaction, creatine kinase. So hopefully that makes sense, that that's the rate limiting enzyme there. Like I'm not going to test you on that, because if there's only one reaction, it's stupid to say anything's rate limiting or not rate limiting, I think, but the book calls it that. Hopefully, like you recall that reaction, stimulators ADP and inhibitors ATP. I would like you to know that. Now for phosphofructokinase, it's functionally the same thing. So AMP, ADP, inorganic phosphates, those are all signs of a low energy environment. So if we have a low energy environment inside of a muscle cell, well, hey, phosphofructokinase needs to start going faster. Now, something interesting here, that pH going up, that means it's getting more basic, less acidic, right? So that, like, just if you're interested, I'm not going to test you on this because this is a hop, skip, and a jump beyond what I want you to know. But the breakdown of ATP and the breakdown of, like, creatine phosphate actually gets us just a hair basic. And there's a reason for that. So, like, ATP, what do we get after we break that down? So ADP, right? And an inorganic phosphate. So something that comes out of that inside of a muscle cell, we get H2PO4 dihydrogen phosphate. So that inorganic phosphate from ATP breakdown, well, that's going to bind to two hydrogens. So that's actually going to make it to where, like in the hydrogen ion concentration, that's what makes us acidic. Hopefully everyone's good with that. So if that inorganic phosphate is binding to some of those, then that's actually going to acutely increase our pH. So like right whenever you start like lifting weights or doing push-ups or whatever you do, your muscle actually gets slightly basic, and then it'll get really acidic after a while. Kind of interesting to anybody? Yeah, I don't know. So like that's a signal there. Now these inhibitors. Let's think about these. For glycolysis, ATP, like CP, that's creatine phosphate. If we have a lot of ATP, a lot of creatine phosphate, PFK, that doesn't need to do anything, right? We're all good. Like we don't need you. Now citrate, something interesting here. Citrate, that is in the citric acid cycle, the Krebs cycle. So whenever we're going through the Krebs cycle too quickly, we actually start exporting citrate out of the mitochondria into the rest of the cell. And that citrate will bind to a certain part of PFK and slow it down. Now, for those of you that are interested in going beyond this, that really only happens in the liver. Like the citrate being exported out, if anybody knows anything about like de novo lipogenesis, like exporting citrate out of the mitochondria, that's like the first step in it, more or less. And we don't do de novo lipogenesis or creation of fat in muscle cells. So like hopefully that's fascinating to some of you. Next thing here, Krebs cycle, isocitrate dehydrogenase, ADP. Don't worry about the calcium ions, but ADP, low energy environment. NAD, that's a stimulator, and NADH, that's an inhibitor. So outcomes of the Krebs cycle are a lot of NADHs, right? So that is going to functionally slow down like isocitrate dehydrogenase if we have too many ADHs around. And then the very last one, the electron transport chain, cytochrome oxidase. Don't worry about knowing that that's the rate limiting enzyme there, because I'm not even sure I believe that. Cool. All right. So all of those stimulators, inhibitors, all sorts of things. All right. Interaction between aerobic and anaerobic ATP production. So here, let's talk about this. So energy to perform exercise comes from an interaction between aerobic and anaerobic, right? So short-term exercise, whenever we have high need, or like not so much high need for ATP, but more so immediate need for ATP. There's a greater contribution like for short-term high-intensity exercise, right? Now for long-term, like, and think about this like sprinting 60 meters, 100 meters, something like that, versus running a marathon. Those are kind of the two ends of the spectrum that I want you to think about. For long-term, low to moderate intensity, almost all of the energy production is actually coming from aerobic sources. And this here more or less shows the interplay between them. So our anaerobic sources of energy, creatine phosphate, hopefully everyone knows that, right? The myokinase reaction, that's another one. But then the anaerobic aspect of glycolysis, so those 4 total ATP or 2 net ATP that we get, that's more or less the sum total of all of our anaerobic sources of energy. So like weightlifting or diving or gymnastics, which are functionally 100% anaerobic, all of that is being supplied by the phosphocreatine system, ATP-PC system, and anaerobic glycolysis. And as we go down, we start getting more and more from the mitochondria. So it actually happens relatively quick. Just about the crossover point seems to be around like 3 minutes or so. So about 50-50, that's around 2.5, 3 minutes, something like that, just about there. So something like these other, something like an 800-meter dash, which typically we think of as being relatively anaerobic, right? Like 800-meter dash, like that normally only takes like, I don't know, 2 minutes or so to run. Like if you're good, I guess. Our like ATP production is around 50-50 from like the cytosolic, like glycolysis, ATP-PC, and also the mitochondria, being the oxidative one, or like aerobic one. And then it goes on and on to where like about like a marathon and longer really is almost entirely aerobic. Is everyone good with this? Right? Now, granted, things like, where is it? Mike Stibor and I, we always talk about like basketball, which like if any of you know anything about me, I don't know anything about basketball. But like we look at the different energy systems of it, and that's around 50-50, I think. I can't really see it. Oh, no, like, yeah, it's saying it's right about there. So right there, I couldn't remember. The thing really about it is it's more or less like position dependent. Does that make sense to everybody? Right? Like, I don't know, a center might do a different thing than a point guard. I'm not going to test you about that to any degree at all. So moving on from there, I think there's just, let's get through just a little bit of this. Oh, yeah, of course. That would happen right there. That's okay. That's So fries, everyone knows, hopefully good, right? But let's get through just like a couple of slides of exercise metabolism. And if you have it pulled up, there's probably a few like repeat slides that I'm gonna go over like super fast. So like this, if you didn't take it before, like short term, high intensity, mostly anaerobic, long term, low intensity, moderate intensity, give or take, majority aerobic. So meaning with oxygen, without oxygen. Hopefully everyone's good with that. Already talked about like how I didn't know where basketball was in terms of anaerobic energy contribution. Now this is something that I don't think you'll have this slide. Oh, you do? Okay, okay. A couple of points I wanted to make about this. Percent of energy contribution for the different energy systems. I want you to write these down. The ATP store, we do store some ATP, and it can produce like the majority of our energy for upwards of half a second to a second. Like if you're looking up here, the red line, it plummets at 2 seconds in. So we actually do just have some ATP as ATP circulating around. Everyone good with that? Right? Now we need to like replenish that through all of the other energy systems. So ATP, CP, that system, and collectively I like to think about this along with the myokinase system. That one is the majority of the energy for around 5 seconds. And typically, this is how I write it, 5 to 15 seconds. So if an individual has been taking creatine monohydrate for a long enough period of time, they will have higher stores of phosphocreatine and be able to extend this a little bit. Is everyone good with that? Right? Everyone good with that? Okay. Now the next one, lactic acid system. This is anaerobic glycolysis, or glycolysis. We can just call it that. But lactic acid being like functionally like the end product of it, if we're actually pushing our intensity enough, it seems like it can be the majority of the energy, upwards of about 90 seconds. So 90 seconds to 3 minutes depending. Now, hopefully you remember, like pyruvate versus lactate. Which one had more hydrogen in it? Lactate. Lactate? Okay, yes. Lactate. So lactate had more hydrogen in it. So functionally, lactic acid is produced to lower the amount of acid inside of a muscle, right? So a couple of things that we could do to extend this energy system, the lactic acid system, would be to take different what are they called? Buffers, like acid buffers. So do all of you all have like baking soda in your fridge? Hopefully, right? So like that accepts sodium bicarbonate is what it is. We actually make some like in our kidneys and stuff. But like if you eat sodium bicarbonate, that can actually make it into a muscle and essentially suck up some of the hydrogen ions so that we can do more anaerobic glycolysis. It actually does seem to help in 400 and 800 meter dashes. But like the problem here is people who do it, like has anyone ever like eaten baking soda before? Like, and I'm not just talking like the teaspoon and cookies. Okay, so I have, right? And you get severe gastrointestinal distress. So it's has anyone ever thrown up in here? Right? Like or had diarrhea, I guess, right? So like all of that, right? It's fun. Like something else that individuals can do for this, and this will be the last thing I'm going to talk about. Does anybody know what like beta-alanine is? Beta-alanine. So beta-alanine is actually converted to something called carnosine, which is functionally just like sodium bicarbonate in the sense that it like accepts hydrogen ions. So if you're taking beta-alanine, the thing that gives you like the tingles in your ears and all that stuff, it actually accepts hydrogen ions and it can extend your lactic acid system just a little bit. And so, like, more or less, if you're taking it for anything that isn't like a 400 or 800 meter dash, it's probably not doing that much. It might actually help you recover between like weightlifting sets, maybe, but there needs to be more research on it. So that's all I have to say. Watch the ice kinetic stuff. I'll see you in lab tomorrow. Rock on.